Hello, everybody. Welcome to Global IMMUNOTALKS. My name's Laura McKay. I'm from the Doherty Institute in Melbourne. And today we have a great global immuno speaker, Dr. Joanna Groom from the Walter and Eliza Hall in Melbourne. And it was my pleasure and honor to introduce Jo to everybody today. But just before I give everybody a bit of background on Jo, I would like to remind everybody that in this episode, this is a pre-record coming from Melbourne, Australia. And so the Q&A will still be conducted on Twitter. I'll share the hashtags on how to do that at the end of the talk. And Jo will be jumping online back on Australia time to answer all of your questions for a little while after the talk. So please jump online and ask all your questions to Dr. Groom after the talk. So next week, we will have Andrea Ablasa joining us. So please tune in again next week. So today, Joanna, we're so happy to have you, introducing you on behalf of our 2022 Global IMMUNOTALK organizers. Dr. Joanna Groom is an associate professor and NHMRC leadership investigator and laboratory head at the Walter and Eliza Hall Institute in Melbourne. Joanna did her PhD at the Garvan Institute in Sydney. Originally, Jo's from Australia. She then went to Mass General Hospital and Harvard Medical School in Boston, where she did her postdoctoral training under the guidance of Andrew Luster. And it's really here that Jo became really well known for her beautiful work on cellular migration and how chemokines can influence T cell fate and function. Jo then moved back to Melbourne following her stint in the States to set up her own laboratory. And here Jo works on all different facets of T cell interactions with various cell types, concentrating largely on T cells. And Jo will present some of the beautiful work from her laboratory today, which we're so excited to hear about. So Jo, before we get into your research, as you may know, part of a tradition here on Global IMMUNOTALKS is that we love the audience to get to know you a bit better. And so we've got a question for you just to give everybody a bit of an insight into your path into science. So the question that we've picked for you is, what trait in your personality helped you most in your scientific career? So I'll just switch over to you, Jo. Thank you so much.
Thanks, Laura. It's really fantastic to join you today. Really enjoyed this these sessions of these talks, and it's nice to be on the other side of the screen for a week. In terms of traits, I think there's probably a few. I think being curiosity driven and just really loving the process of discovery is one of the key drivers for me, as I know it is for a lot of scientists. But within that, I have a real visual focus, and I guess that's why I have such an affinity for imaging methods. But even more than that, sketching out different models and systems, trying to fit pieces of the puzzle together in new ways and identify different gaps in the literature has, I think, really provided me a unique scope to find interesting aspects of the work to uncover. And I guess I would say the counterpoint to being really curious is that you can't spread yourself too thinly. So I guess it's really critical to find that balance between maintaining the curiosity, but also really making sure that you don't spread yourself too thinly.
Thank you, Jo. I think the trainees will really appreciate that advice as well. Even though we love seeing research as a puzzle, yeah, you've got to pick your questions just to keep you on track. Thanks so much, Jo. So we're so looking forward to your talk. Would you like to share your slides? And we'd love to hear from you.
I will start sharing. Thanks, Laura. So if you could just let me know if that's working.
Yep, that's perfect, Jo.
Excellent. And I will find my pointer and really get started. So today I thought that I would start by reflecting on this characteristic textbook representation of the adaptive immune response. And this curve really focuses our attention on the magnitude of response. And we know that this can be measured in a lot of different ways, from looking at antibody production to the individual cell numbers of our adaptive immune cells, our T and B cells. And in my lab, as Laura said, we're really interested in dissecting T cell responses. So from that perspective, this cascade really kicks off when a T cell interacts with a dendritic cell. And this leads to a huge proliferation of these cells and differentiation. And following a pathogen such as a viral infection, which is a model that we study a lot, we differentiate our naive T cells into short-lived effector cells. And these are responsible for clearing a viral infection. And following the clearance of the pathogen, this leaves us with a pool of longer-lived memory cells. And it's really these memory cells that hold the power of our adaptive immune system. Because we know that when they see a secondary challenge, they respond more rapidly and with greater intensity. So when we understand this curve, we can really harness this potential. And I guess the best known example of this is through vaccination, something that's really at the forefront of many of our minds at the moment. So we know, and we've seen really beautiful examples through the Global IMMUNOTALK series, different ways that we can study this curve and really unpack its inner workings. But for my lab, we look at this through a lens of T-cell interactions. So I guess what interactions are we interested in particular? Well, we really consider many interactions through the life of a T-cell, from that point where they land in secondary lymphoid tissue. So that means, first of all, right at the start, following an initial exposure of a pathogen, when T cells first meet their antigen in the context of dendritic cell presentation. And we know that this can be performed by different dendritic cell subsets, and that this interaction really determines T cell fate. Later, during the peak of the adaptive immune response, we switch our attention to now focus on interactions within the germinal center. And so these are interactions between now CD4 T cells and germinal center B cells. And we know that these lead to highly protective antibody production. And what we're really interested in understanding here is how these interactions are altered depending on the context of the infection. And then lastly, following clearance of a pathogen, we're interested in how T cells are positioning within secondary lymphoid organs and how this impacts their ability to form that long-lived pool of memory. So I just wanted to go through, I guess, two stories from the lab that kind of highlight these different cellular interactions that we're interested in studying. The first, we look at CD4 T cell interactions and how this differentiation is distinct in different pathogen infections and really is tuned towards different responses in these settings. And secondly, one of the key aims of the lab is how we make better CD8 T cell memory. And for this study, we really took about trying to understand whereabouts that differentiation process takes place. So firstly, we'll start with this story that was led by a PhD student in the lab, Amanya Sheikh, and really, as I said, looks at how T-cell differentiation is tuned in different settings and how this in turn has an impact on our germinal center responses and production of antibodies. So we really consider T follicular helper cells as gatekeepers for the germinal center response. And when I say that, I mean that we have as TFH cell number increases, we also have an increase in the number of germinal center B cells. And one thing I find intriguing about T follicular helper cells is really their breadth of responses. So they're involved in many different settings that we encounter, from vaccine response to pathogen challenges, including viral, bacterial, Helminth and fungal infection. They've also been shown to have a protective role during cancer, and in particular with checkpoint inhibition. So this is really expansive tailoring of these cells during these different settings. And I guess the counter side of this, when we think of them as gatekeepers of germinal center reactions, is when there's dysregulation, that can lead to auto-antibody production, such as we see in autoimmunity, such as lupus or rheumatoid arthritis, but also other antibody-mediated diseases, such as asthma and allergy. So this real breadth of function really sets these cells apart from how we generally consider CD4 T cell differentiation and specialization. So we're all probably very familiar with this scheme where we have our CD4 effectors, Th1, Th2, and Th17 effectors. And the differentiation to each of these is really guided by a set of lineage-defining transcription factors and specialized effector cytokines that give them the ability to have a very set function with different infectious and inflammatory challenges. So T follicular helper cells really, and also T regulatory cells, stand really quite apart from this paradigm because they can differentiate in all of these settings that we see above. And so what we started to think about in the lab is really how this is orchestrated and how it is that our T follicular helper cells are actually differentiating in parallel to our other effector subsets. And so for this, we started to focus on viral infection and started to look really at the role of the lineage defining factor of Th1 cells, T-bet, and how this might impact the formation of TFH cells. Now, we know from previous work that there's a counter regulation in these transcriptional regulators such that they really repress alternate fates. So we were interested if the inflammatory milieu was leading to T-bet being expressed in TFH cells during viral infection. And so for this study, we started with these beautiful ZS green T-bet reporter mice. And here we have an influenza infection, and we're looking at the draining mediastinal lymph node at day 4, 8, and 14 following infection. And we can see even at the very start of the germinal center response that we have T-bet positive cells that really seed the germinal center. And when we have an established germinal center, we can see those cells very nicely in the light zone of the germinal center. We went on to show that these were in fact T follicular helper cells in this position. So from this, it's quite clear that our TFH cells can express T-bet in this setting. So we went on to really track how the levels of T-bet expression using this reporter, and here you can see the setup with TFH cells in red and TH1 cells as a comparison in blue. And the expression of T-bet increases in both of these subsets in parallel during the course of the infection. And we can see a graded expression of T-bet for both our TH1 and TFH cells. Now, our TH1 cells have a very high level of T-bet expression. And by comparison, our TFH cells have a more moderate to intermediate level of expression, but quite a clear population of T-bet expressors. And the kinetics of this really track very in a similar manner between the two subsets. So then what is the role of T-bet during TFH differentiation? Well, using this system, we used a conditional knockout of T-bet within the T cell compartment. And we could see here, again, following influenza infection, the expected decrease by frequency and total cell numbers, decrease in TH1 cells. And in contrast, we saw this increase in the frequency and total cell number of T follicular helper cells. So here we're looking at the polyclonal populations, and we went on to use MHC+2 tetramers to identify antigen-specific epitopes in this system. And again, we could confirm that decrease in TH1 cells and increase in TFH cells. So more than just looking at cell number, we were interested to understand how these cells were changing in their function. And to look at that, we started to investigate their cytokine profiles. And so here we're using IL-21 reporter mouse. And while we didn't see any change in the total amount of IL-21 cells, what we did see is that our TFH cells now that are deficient in T-bet expression can no longer express interferon gamma. And this means that we completely lose this population of polyfunctional TFH cells. So what does that mean, I guess, for the B cells that these cells are talking to? When we looked at the isotypes that these B cells were producing, now we're looking at an intact T-bet deficient mouse. We could see an increase in IgG1 production and a decrease in IgG2AC. And we could see this not only by looking at the cell surface antibody within our germinal center B cell compartment, but we could also see this in the antibody levels in the serum of these mice. Again, an increase in IgG1 and a decrease in IgG2AC. So then we turned to our cell type specific knockouts to really unpack the different the cellular requirements for these isotype switching. And we could see here in the T cell specific knockout that this was entirely responsible for that increase of IgG1, but it didn't play a role in the switching towards IgG2AC. And we went on to show with a B cell specific T-bet deficient mouse that was a B cell intrinsic role. So what this work was showing us was that T-bet acts as a differentiation switch between TH1 cells and TFH cells during influenza infection. And this really fit very nicely with previous work showing this counter-regulation of T-bet in other effector subsets, such as Th2 differentiation. But while Emmanuel was collecting this data, another paper was published that really threw us a little bit. And here, this group, Jason Weinman's group, looked at the role of T-bet during TFH differentiation, but now choosing a different viral infection, choosing LCMV, whereas we were previously looking at influenza. And they showed something really distinct, that whereas a switch between these two subsets, now T-bet was absolutely required for the formation of both Th1 and TFH cells. So I guess as any self-respecting scientist would do when they encounter work that doesn't fit with their hypothesis, is really to try and replicate those experiments in their own hands to see if it holds true. And so that's exactly what we did. And we can see here now we've crossed our T-bet deficient mice onto the smarter allele, and this allows us to track CD4 antigen-specific cells following LCMV. And when we do this, we can see that decrease in Th1 and that decrease in the formation of TFH cells. So in this setting, following LCMV infection, these cells are really both require T-bet expression. So this is really curious, right? Because we've got two very classical models of viral infection, and yet the lineage defining factor of Th1 cells, T-bet, has very distinct roles for the formation of TFH cells. So we started characterizing what the differences between these two, between T cells in these two different settings. And we started to return to our T-bet reporter. And what we saw was that in LCMV, there was a much higher induction of T-bet, not only in our Th1 cells, but also in the TFH cells. And this was true early in infection at day 4 for both infections, and also at day 8, where we could really see a threshold level of T-bet expression, where in cell types such as both Th1 populations in either infection or in TFH cells in LCMV, when T-bet was increased to a certain level, those cells completely relied on T-bet expression for their differentiation and survival. So in characterizing the very early T cell differentiation states between these two systems, we noted a really striking difference in the expression level of CD25. And this was quite interesting to us because there had previously been described a paradigm for the production and consumption of IL-2 between TFH cells and Th1 cells. And so this fits that IL-2 production from TFH cells in turn is consumed by Th1 cells based on their expression of the IL-2 high affinity receptor, CD25. And so this provides, I guess, a theory as to how these two cell types can differentiate in parallel. And so we looked at this in both of these systems. And when we looked at LCMV, we could see this clear separation between these distinct fates, where again, very early on following infection, our pre-TFH cells were marked by their expression of CXCR5, and these were very distinct from our IL-2 consumers, the precursors of Th1 cells. And again, when we looked in LCMV at the production of IL-2 in this setting, This was, again, extremely distinct populations early in infection where we had a high level of IL-2 production, which was very distinct from the population expressing and consuming this IL-2. So it seemed that in this infection, this paradigm really holds up very well. However, when we compared this with what we see in influenza, we didn't have these distinction of fates at this time, and there was far less IL-2 being produced in this setting. And so we think that this IL-2 was setting that threshold for T-bet expression between these two infections. So this really shows that the formation of TFH cells occurs in a very context-dependent role. And this is important when we consider that breadth of functions that we see for TFH within TFH diversity. So this study demonstrates that distinct environmental settings regulate the transcriptional differentiation of TFH cells. And in turn, we're now looking at how, when the differentiation process is altered, how this really affects the function of these cells in a wide range of settings. So now I'll turn to, I guess, the other arm of what my lab works on, CD8 T cell biology, with this question of how we can generate better memory cells. So this project has been led by another PhD student in the lab, Bridget Duckworth, and it was initiated through a collaboration with a previous postdoc, Fanny LaForest and Verena Wimmer in our WeHi's dynamic imaging facility. So we started with this question of how a naive T cell differentiates into either an effector, short-lived effector cell, or a memory precursor, and whereabouts this actually takes place. So this decision is known to take place in secondary lymphoid organs, such as the lymph node or the spleen, but we're very interested in looking at the structure of lymph nodes for this question. So these are highly organized structures, and this structure actually plays a very key role in the activation of T cells and their differentiation responses. And so I'll just guide you through the different niches that we see in a lymph node. We have in the center this T cell paracortex, and this is where naive T cells accumulate and interact with dendritic cells and scan dendritic cells in search of antigen. And then we have our B cell follicles around the outside of the lymph node, and between each follicle have these interfollicular regions that form pockets between each of the follicles. So movement of T cells and other immune cells around these distinct niches of the lymph node is guided by small chemoattracting molecules known as chemokines. So we kind of started this story in a very similar place. We know that T-bet marks CD8 T cells now as the first transcriptional readout of their effector potential. And so we took these same ZS green T-bet mice and we crossed them now onto a P14 TCR transgenic, and this allowed us to track the expression of T-bet in an antigen-specific way following LCMV infection. And you can see here in a time course the upregulation of T-bet, and this occurs prior to these cells becoming effectors, which is marked by their expression of KLRG1. So we were interested in where this process takes place. So for this, following infection, we cleared these nodes using a method called CD3D, which has been developed by Michael Gerner and Ron Germain. And we took our cleared nodes and we imaged them using light sheet microscopy. So what you can see here, this is a day 3 node following an infection, and we have all of our P14 cells marked in DS red and our ZS green reporter. And what you can see is the very earliest point where we could start to see expression of T-bet. These cells are already located in these interfollicular pockets in the periphery of our lymph node. And we could confirm this with confocal staining, showing that indeed these cells out in these interfollicular regions were the ones that were co-staining for KLRG1, as opposed to what we could see in the T cell paracortex. So we realized quite quickly when we started this method that we needed to develop a new way to quantify T cell location in 3-dimensional space. And so for this, we adapted an ImageJ script, which allowed us to segregate our lymph node into 100 different layers of equal volume, from the periphery to the center of the lymph node, again in 3 dimensions. And then we could overlay our cellular objects, our T cells, onto this map. And this allowed us to give a density score of the location of T cells throughout the lymph node. So when we do that, we generate a graph that looks like this. So here now we kind of have flattened that 3-dimensional lymph node out to look at the periphery, here marked as 0, and the center of the lymph node marked as 1. And we can see our cells distributed over throughout the lymph node. And here we have our p-value line for the differences that we see between 2 populations. So we started here quantifying the position of total P14 cells, CD8 cells in this system, and we could see there was a very nice bimodal distribution where most cells were located in the center of the lymph node and some were located out in this periphery. But when we just quantified those cells with very high expression of T-bet, we could see that these cells were really skewed towards the periphery of the lymph node out in those interfollicular regions that we had observed. And using a range of methods, we honed in on a few regions of interest within the lymph node. So this region in the periphery, it relates to our interfollicular zone, and this region in the center relates to the within the T cell paracortex. And again, we could see that those T-bet high cells were located quite significantly more in the periphery. So next we wanted to understand how these effectors were moving into this area. And CXCR3, the chemokine receptor, was a top candidate for this for several reasons, but mostly because CXCR3 is regulated by T-bet. So just to give you a little bit of background, CXCR3, as I said, is a chemokine receptor. It binds 3 ligands, but in particular, we're interested in looking at the biology of CXCL9 and CXCL10. And that's because CXCL11 has several point mutations in black 6 mice, so they don't produce a functional chemokine in that strain, which is the strain that we generally use. And more than that, we've recently conducted a study where we've corrected those mutations using CRISPR-Cas9, and we found that CXCL11 didn't really have a role in this setting. So as I said, CXCR3 is regulated by the expression of T-bet, and we can see that very nicely here, this co-expression of both the T-bet reporter and CXCR3 over the time course of LCMV infection. So now we wanted to compare the positioning of cells, either wild type cells or CXCR3 cells following infection. So now we're looking again at our intact nodes and comparing the positioning between our wild type and our CXCR3 knockouts. And we see in our wild type setting that same movement out into the peripheral regions of the lymph node quite clearly, with some cells still being found in the central paracortex, but a huge migration out into these interfollicular regions. And in contrast, in the CXCR3 knockout, these cells were much more retained in the central paracortex and less likely to be positioned out in these interfollicular regions. So now we just have some longitudinal clipping slices here through the center of these lymph nodes, just to emphasize the difference in the positioning between these two settings. And when we quantify this, again, we can see that the CXCR3 knockout cells are much more retained in the center of the lymph node compared to the wild types. And we can see that in particular here in the T cell paracortex. So these cells are retained in the center of the lymph node, but what does that actually mean in terms of their potential to form different effector populations? So when we looked at this, we saw that the CXCR3 T cells had lacked an ability to form CD8 effectors, marked here again by the expression of KLRG1. And we found a reciprocal increase in the ability of these cells to become memory cells. And we searched a large range of memory markers, looking at different subsets of CD8 memory cells. And quite interestingly, we found that it wasn't all central memory cells that were increased when we knock out CXCR3. And in fact, it was a very specific increase in a population of memory cells named stem-like memory cells. And these cells, as their name suggests, are pre-programmed for self-renewal, and they repress terminal differentiation. And it's been shown that these cells provide the proliferative burst following cancer immunotherapy. And so understanding specifically how these cells differentiate really has potential therapeutic benefit. And so just moving to this model, we showed that these effector cells move out into the periphery of the lymph node, and this is guided really by the combination of the transcriptional regulator T-bet and expression of CXCR3. And in contrast, we have differentiation of stem-like memory cells from those cells that are retained in the center of the lymph node. Now, these cells are marked by a few different markers, including CD62L and the transcriptional regulator TCF1, and other cell surface markers such as SLAMF6. So next, we wanted to understand what was driving this differential positioning. And so for this, we turned to look at these dual chemokine reporters. Now, these are known as REX3 transgenic mice, and they're a mouse model that I generated when I was in Andy Luster's lab at Mass General. And so here we report the expression of CXCR9 in magenta and CXCR10 in cyan within the single mouse. So following infection, we cleared these lymph nodes again. And what was really striking is that these different chemokine ligands were expressed in distinct locations within the lymph node, so that we could see CXCR10 being expressed in a dominant way out in these interfollicular regions. And CXCR9 had a much more homogeneous expression throughout the lymph node, but with far more cells being expressed, expressing CXCR9 in the center of the lymph node. And so we can see that quantification over here. We then looked to understand what the cells that were specifically producing both of these ligands were. And we found a sizable contribution to our stromal cell niche, but also our dendritic cell populations. And interestingly, we could see a clear distinction in the potential of chemokine expression between our different conventional dendritic cell populations. So CDC1 cells produce predominantly CXCR9, and we tracked down this location to indeed be in the center of the lymph node. And conventional type 2 dendritic cells express far more CXCR10. And again, we located these cells out in the periphery. And so this allowed us to really match up those cellular partners for our T cells in each of these distinct lymph node niches. So in keeping with that expression out in the periphery of CXCR10, when we now used the chemokine ligands knockout models as the hosts for our experiments, we could show that it was indeed CXCR10 that was moving cells out into the periphery of the lymph node. And when we knocked out CXCR10, these cells were much more restricted to the T cell paracortex. And to reinforce that link between location and differentiation, these cells also had a much higher potential to become stem-like memory cells. So now we could fill in this model a few steps further, where we have our effector cells here out in the interfollicular area regions interacting with our CXCR10 producing CDC2 cells. But what this didn't allow us to answer was what were the factors that were retaining our stem-like memory cells out in the center of our lymph node. And when we first started thinking about this, we became really interested in this relationship between the transcriptional regulator and how it upregulates the expression of CXCR3. And we, I guess, hypothesized that potentially TCF1 as the defining factor of these stem-like memory cells might be acting in a very similar way and so we wanted to identify if it was regulating any chemokine receptors. So first of all, we looked at CXCR3 expression between these two populations, and we couldn't see a difference. And indeed, when we look in a migration assay here, there was no difference in the potential of either effectors or stem-like memory cells to actually migrate towards CXCR10. And this was obviously lost in the knockout population of each of these subsets. So we turned to TCF1 ChIP-seq to identify some TCF binding sites within the gene for CCR7, another chemokine receptor that has been shown to regulate the position of T cells and dendritic cells into the paracortex of lymph nodes. And then we looked again at RNA-seq data when we lack TCF1 to strengthen this concept that TCF1 is a direct regulator of CCR7 expression. And to confirm this in a really functional way, we went back to our in vitro migration assay, and indeed we could see that our stem-like memory cells had a greater potential to migrate to one of the CCR7 ligands, CCL21, and that this was not at all diminished when we lack CXCR3. And so coming to our model, we can see this close relationship between the transcriptional regulation and the regulation of chemokine receptors. And this is true now not only for what we see in our effector population, but also for the formation of stem-like memory cells. So finally, we really wanted to test this relationship between the location of cells and their differentiation potential. And so for this, we wanted to take a different approach to identify other factors that might modulate this, the location of cells in vivo. And we turned to the type 1 interferon family and looked at the role of the type 1 interferon receptor in this system. Now we know that CXCR10 in particular is regulated by type 1 interferons, and so we propose that this is acting upstream of the induction of chemokines in this setting. And so when we compared now our wild type and our IFNAR knockouts, again, we see these cell accumulation in the center of these lymph nodes compared to the wild type hosts. And this is seen very clearly here when we quantify. And again, this difference in location fed back into the potential differentiation of these cells, where we have a loss of effector formation and an increase in their stem-like memory potential. So to summarize, this study really shows that T-cell fate is imprinted in distinct lymph node niches. And this can be extrinsically tuned by the deficiency in CXCR10 or type 1 interferon signaling in order to promote stem-like memory differentiation. And what this work has done is brought us back to reevaluate how we look at this curve. So as I said, this really focuses our attention on the magnitude of the overall immune response. However, this work highlights that this is in fact the sum of the magnitudes of both the effector response and the magnitude of the memory response, and that these are two quite distinct processes that occur simultaneously within our secondary lymphoid organs. So we've become very interested in how these different modules are regulated. So we think that this is quite important insight, because when we think about effector cells and memory cells, actually they have two very distinct purposes, either to immediately eradicate an ongoing challenge or to preserve themselves for a challenge in the future. And our work, along with the work of others, suggests that there's a real tension between these two fates, such that when we block one, we can enhance the other, and that cell location and the environment of these cells really provides us some nice leverage between these two fates. And then lastly, to bring this together, we see some synergy between these differentiation processes, not just for CD8 cells, but now also for CD4 cells, where this regulation between the transcriptional signature of these cells is intertwined with the location of these cells and the environments that they find themselves in. And so we're very interested in understanding how both of these modules are regulated for both CD4 T cells and CD8 cells, and exploring ways where we can not only tune efficient TFH cells, but also making and generating better CD8 T cell memory. So that just leaves me to acknowledge and thank the people, particularly in my lab, who've contributed to this work. As I said, highlighted the work of Amanya and Bridget in the lab, and the rest of the lab has really supported them and have projects that continue to look at different aspects of both of these projects. And to thank particularly the WeHi dynamic imaging facility that we work incredibly closely with, and also our bioinformatician collaborators and other collaborators, not just at WeHi, but throughout Melbourne. So thank you. And I look forward to getting your questions on Twitter.
Thank you so much for that, Jo. That was such a beautiful overview. I enjoyed that so much. I'm sure everybody will when this talk goes online. So just to let everybody know how to do that Q&A, I will just share a slide on how to do this. Right, just switching over now. Okay, so to get in touch with Jo and ask all your questions about her wonderful talk, please go to Twitter and search for the account Global IMMUNOTALKS. Find the tweet that says ask questions for Dr. Joanna Groom here, and then Jo will reply to that tweet using her own account at Groom Lab, and you will reply to your tweets with your question mentioning the hashtag Global Immuno. And Jo will be willing to do that at least for the next week or so. So please get in touch. Thank you so much, everybody, for joining us. Thank you again, Jo, for such an incredible talk. We'll all be enjoying that for so much when that's uploaded to YouTube. And we'll see everybody again for another Global Immunotalk next week. Thank you so much.
Thank you.